In this video, we're going to look at organelles, and we're going to look at the membrane-bound organelles that are found only in eukaryotes. Now, to remind ourselves, the last video and on the last page of notes, all those structures, things like ribosomes, DNA, cytoplasm, both eukaryotes and prokaryotes have those, right? Bacteria, as well as plants and animal cells, have those. But for this video, the notes you'll be taking today, these structures are not found in prokaryotes. Things like nucleus, rough and smooth ER, Golgi Apparatus, Mitochondria, lysosomes, these are not found in prokaryotes, only in eukaryotes. This video is going to be very important because there'll be a lot of exam questions coming from it, but it's also important if you're one of my honor students because I'm going to give you some pointers as we go through the video as you're making your project, like, hey, this organelle should look this way, or so forth. I'm going to give you a bunch of hints, so very important video. Think of organelles. The term organelle actually means little organ. You should write that in somewhere so you can kind of remind yourself about that. What are our organs? Well, we have a body, but that body is made of different parts. We have a nervous system with an organ called the brain that's controlling everything through nerve impulses. We have a special organ called the heart that pumps blood. We have special organs called lungs that obtain oxygen from the air and put it into our blood. We've got the different organs of the digestive system. And so these organs have specific functions, and they're working all day long to do what they do and help us function overall. All these different organs are doing their own little piece of the puzzle. In much the same way, on a cellular level, we have these different organelles. Each of them has their own specialized function. They're doing all these functions all the time, and as a result, these eukaryotic cells, like the cells we're made of, are more complicated, and they can do more varieties of functions because of the diversity of these organelles inside of them. Plant and animal cells, if you take a look at the picture here, these two eukaryotic cells, you'll see that they're pretty similar in a lot of ways. They both have a lot of the same organelles. However, you'll notice that there are a few organelles that are different. And so it's important that you keep track of that too as you're making your project, again, if you're one of my honor students. The first one is actually kind of an outlier because it's not a membrane-bound organelle. It's a structure, though, that's not found in prokaryotes because it's a channel that connects cells together, and because prokaryotes are only unicellular, of course, they don't have those things. They're found only in plant cells, and they're called Plasmodesmata. What they are little tunnels that connect the cells of a plant. And so you can see this picture here. This is a really awesome photograph here with an electron microscope. And so you can see that these little channels that connect them. You got the cell walls there. And then you can see that some of these ones that look more faded. Imagine those are ones that are more kind of in the background or the foreground, right? Because this is a two-dimensional photo of a three-dimensional world. So pretty cool. In real life, understand that a lot of our cells are actually connected by kind of similar structures, but we don't need to go into all those details. That's more for like a cell biology class in college. For now, I just want you to know about these ones. These are found in plant cells. They're very important for plants, and they're called Plasmodesmata. This next one is also not technically a membrane-bound organelle, but it is an important organelle, and this one's found only in animal cells, so not in plant cells. It's called the centrioles, and these are structures made of microtubules. So on your project, if you're doing a plant cell, you shouldn't have these. If you're doing an animal cell, they should be made out of the same structures as you used for the microtubule. So you can see, each one of these looks like a microtubule, and they're attached together, and then you get this overall structure called a centriole. They function during cell division. The other thing that's kind of interesting about them is that we always find them in a T shape. So in other words, you have one that's kind of lying this way, and then you have another one that's lying perpendicular to it. Now, the last I checked, we didn't actually know exactly why that was, or what important feature that might cause. But the reality is we always see them in this letter T shape, and so if you're doing an animal cell, your centriole should be in that shape. Here's a beautiful picture that shows it. There's a scanning electron microscope image, so you can see that we're looking down at one, right? You can see these beautiful picture that shows the resolution. You can actually see these microtubules like that. And then the next one is lying perpendicular to it. Again, so it'd be kind of like a T shape, just like I drew on the screen there earlier, right? So you've got one that's laying this way, like that, and you have another one that's perpendicular to it, right? That's kind of standing straight up, and we're looking down it, like that one. So that's what the centrioles are, function during cell division. If you're wondering why don't plant cells have them, well, plant cells can do cell division a little bit differently because they have cell walls, and so that they don't really need these centrioles the way that animal cells do. We'll get into more detail about centrioles when we're talking about cell division in the mitosis unit, which is coming up in a few units. The next two organelles are going to have important shapes and colors. So the first one is chloroplasts, and they're green, and they've got an interesting internal shape that we'll talk about in a second. What I need you to know about chloroplasts, though, is their function, very, very important. They carry out photosynthesis. And what photosynthesis is that they convert light energy into chemical energy in the form of sugar, glucose. So these are the organelles that we find a lot of in the green leaves that are sort of the solar panels, capturing that solar energy, turning it into sugar energy. Now, they are found primarily in plant cells. Just to be clear, for my class, we're going to kind of turn that primarily. We're just going to turn that into an only. Okay? Now, the reason that I say primarily, though, in real life is there actually are some animal cells that are the exception to the rule. There are some animal cells that do have chloroplasts, and they can do both photosynthesis and also consume other organisms. These are all microscopic organisms that be doing this. But 99.99% of the time, animal cells don't have chloroplasts and plant cells do. So if you're making an animal model, for example, better not include chloroplasts. And if you're doing a plant cell, you better include chloroplasts. Now, if you look at the internal structure, you'll see that it's very interesting. You have this overall organelle, and then inside of it are these stacks of these. They look almost like pancakes. And the stacks, they're called granum, which are stacks of these little pancake-looking things that are called thylakoids. And we'll learn in the next unit what's actually happening at these different regions of the chloroplast. Now, how in the world do we know that? Well, if you look at the cell, this is what we'll see under the microscope in this unit. You can see the cell wall of this plant cell, and you can see all these green dots are the chloroplasts. And you'll notice that's the only part of the plant that's actually green. So when you look at a green leaf, that is caused by these green chloroplasts. Here's another picture. There's a scanning electron microscope where you can see the cell's been cut in half, and you can see the chloroplasts inside of it. And then here's a beautiful picture. Look at this. This right here is a chloroplast. So this kind of outer green line is the membrane that surrounds the chloroplast. And then inside of it, you can see these stacks. These are those thylakoids that are stacked up. And again, next unit we'll actually see what's happening at these different regions of the chloroplast. But for now, I just want you to know that they have this important internal structure. And if you're doing a model, if you're one of my honor students, I would really very much like to see that internal structure illustrated somehow in your model. Plus, this is one of the few organelles that better be a specific color. These ones better be green, because that's what the color they are in real life. The next one then is mitochondria, and they have a very distinct color as well. They're sort of a deep brown, almost bordering on red kind of color. They're doing something important too. They're doing something called cellular respiration, which is essentially burning sugar into ATP, which is the cellular energy that living things are using. So the mitochondria are critically important, and notice they have this internal structure as well that's interesting. They have this folded inner membrane inside of them, creating kind of this empty space, which we'll learn about is called the matrix, and we'll learn about all that's happening in here in the next unit when we look at photosynthesis and cellular respiration. Now, their color, though, is a deep brownish red color. Little side note, just kind of for fun. If you think about chickens, when you eat chicken or turkey, but especially chicken, you can know about dark meat and light meat. Now, why do they actually have different colors? Well, the answer is there's actually a couple of reasons, but one of the reasons, is kind of a cool one, is that cells that are highly active, that are using a lot of energy, they're going to need a lot of these cells because they need a lot of these powerhouses, if you will, that are burning through the sugar and producing the energy. So in parts of the chicken that are very energetic, that are using a lot of, the muscles that are actually being active a lot, they're going to have lots of mitochondria, and so they're going to be a darker color. So think about, where's the white meat on a chicken? Well, like a chicken breast is pretty much pure white meat. Well, that's not a very active part of the chicken, right? Chickens don't fly, and they don't do push-ups, right? So they're not using that breast meat very much. What parts of chickens are brown meat, the darker meat? Well, like the legs. Are chickens on their legs all the time? Yeah, they're running around on their legs all the time. So that muscle is much more active, and it's going to have much more mitochondria. Now, there are some other reasons for the color difference, which we don't have to get into, but that's one that's kind of cool, that relates to what we're doing in class. And again, we know that internal structure because we can see with our electron microscopes. We can see that these mitochondria have this distinct inner structure, this folded outer membrane that creates this inner empty space. So this is another one that I expect to be a specific color. And you'll be having mitochondria in both plants and animal cells. That's important to understand too, because both plants and animals need to burn sugar to make ATP. The question is going to be, where do they get the sugar from? Organisms that are made from animal cells, like me, because we don't have chloroplasts, we can't make our own sugar. I have to eat sugar, and I'm going to burn it. Plant cells, because they have chloroplasts, they can make their own sugar in the chloroplasts, and they still have to burn it, so they still need mitochondria. Another fun little kind of side note here that's also important and will be on your exam. Mitochondria and chloroplasts are the only organelles besides the nucleus that have some DNA. They don't have very much, but they have a little bit. So understand that, when you think about like the DNA that makes me who I am, that DNA is housed in the nucleus. But I have a little bit of DNA in my mitochondria as well. And we'll talk about why that is a little bit later on this year. Here's another beautiful picture. And if you think to yourself for a minute, what kind of microscope took this picture? Well, I've got my magnification there. So that's well above my light microscope range, right? And you can see the picture is three-dimensional. So this would be a scanning electron microscope. You can see the outer membrane of the mitochondria. You can see that beautiful folded inner membrane with that empty space inside. Next organelle is an important one, the nucleus. The nucleus is the largest organelle in your project. It should be, pretty much the largest. It's a large organelle that houses the DNA inside of it. It is surrounded by a membrane like all of our membrane-bound organelles, but this one's kind of interesting. It's kind of a double membrane, which you can see right here in the picture. There's sort of two layers of it, and it has its own name. It's called the nuclear envelope. So the nuclear envelope is what surrounds the nucleus and sort of gives it its shape, and then inside of there is the DNA. There are openings inside this nuclear envelope, and those openings are called nuclear pores. That's an important term for you to know as well. And again, if you're an honor student and you're doing your project, your nucleus should be very large, and it should have these pores. I don't know how you want to do it. There's lots of ways you can, but there better be these openings in it that are called nuclear pores. Here's a picture, again, if you think about what kind of microscope took it. We're well over 1000x, so it's an electron microscope, and it's three-dimensional. It's a scanning electron microscope. We're looking at the surface of our nucleus, so this would be the nuclear envelope, and then you can see these openings. These openings are called nuclear pores, and they're allowing materials into and out of the nucleus. The nucleolus then is inside of the nucleus, and it is a region that manufactures ribosomes. So it makes the ribosomes, which if you remember from the other video, ribosomes, their job is to make proteins. Now, what I want you to understand, if you are doing your project here, if you're an honor student, the nucleolus is essentially a massive cluster of ribosomes. So whatever you use, whatever material you use to make your ribosomes, what your nucleolus should be is just a whole bunch of those all stacked together, because the nucleolus is essentially just a massive pile of ribosomes where the ribosomes are being manufactured. Here's a picture. We have two of them here. Both of them are over 1000, so that tells us they're not done with light microscopes, but both of them are two-dimensional. Yeah, they're not three-dimensional, so this would be our transmission electron microscope. You can see, so the cell kind of goes beyond the frame for both these pictures, but you can see quite clearly our nuclear envelope. You can see then the DNA would be in here, right, in here, and then you've got the nucleolus in both of these. Moving on. Another fairly large organelle that should take up a good amount of space in your model, if you're doing this as an honor student, is the endoplasmic reticulum, also abbreviated as ER. The endoplasmic reticulum is a folded membrane system, so it's just a bunch of folded membranes back and forth. And what happens here are two things. You have two things going on here. Number one, you've got proteins being synthesized, and you've also got lipids being synthesized. So this is a major area where the cell is building proteins it needs and lipids that it needs. Now don't get confused here. We know that ribosomes manufacture proteins, right? We know this. But a lot of those ribosomes, if you remember from the last video, they're located in two places. Some of the ribosomes are just kind of randomly in the cytoplasm. Other ribosomes are located on the rough ER. The smooth ER is called that because if you look at it, imagine I could reach out and touch this. Do you see how it looked like it'd be smooth? There's no ribosomes on its surface. So the smooth does not have any ribosomes on it. The rough ER, when you're making your model, it should look exactly like the smooth ER, but then you should take whatever it is that you're making your ribosomes out of and put them all over the surface of the rough ER. So it looks like if I could reach out and touch it, it'd feel rough. Now, because it has lots of ribosomes on it, and those ribosomes are making proteins, the rough ER is a place where we're making lots of proteins. The smooth ER, though, is where we're making lots of lipids. So together, the smooth and rough ER are making proteins, they're making lipids. Here's a picture of what it looks like. So this is a scanning electron microscope image. We can tell because it's over 1,000 and it's 3D. What we're looking at, this is the surface of the endoplasmic reticulum. Does that look smooth or rough to you? You're hopefully saying that it looks rough. These are all the little ribosomes. All these little tiny dots are the ribosomes all over the surface. So this would be the rough ER. If you could imagine, if I was showing you a picture of smooth, how would it compare? Well, it would look exactly the same, but all these ribosomes would be gone, and so we would see it as smooth in appearance. Next, we have the Golgi Apparatus, which is a series of sort of almost looks like pancakes, these flattened membranes. What's happening at the Golgi Apparatus, think of the Golgi Apparatus like the post office. The post office. Now, what happens at the post office? Well, packages, right? They arrive at the post office. The post office may modify them in some way, right? And then the post office ships it to where it needs to go. Yeah, that's what the post office does. So what is the Golgi Apparatus doing? Well, the Golgi Apparatus is receiving packages. It's modifying those packages in some way, and then it's shipping them on. Now, what are the packages? Well, the packages are like the vesicles. So the vesicles are the packages that transport materials to this Golgi Apparatus, and then the Golgi Apparatus modifies what's inside those vesicles and then ships them on. Now, if you're trying to think about what a vesicle is, in real life they look not square, but they look like spheres. So a vesicle is, imagine a sphere, a small sphere of plasma membrane with an opening on the inside, and in that opening you can store stuff. And so that's the package that's being delivered to the Golgi Apparatus. The Golgi Apparatus then is sorting it, modifying it, and moving it on to the next place. When I saw this picture for the first time, I was really blown away because it's just so beautiful when you actually think about what you're seeing. First of all, what kind of microscope took it? Well, it's over 1,000, so we know it's an electron microscope. It's two-dimensional, so it's transmission electron microscope. Then they've gone through and colored it so we can see what we're looking at. Now imagine we're zoomed in on the cell, so realize the cell extends way beyond the picture here. What I'm looking at over here then, this is the nucleus. This is the nucleus. So this would be the DNA in here. What do you think this structure is inside the nucleus? Hopefully you're saying that's the nucleolus. What's this structure here? That's the nuclear envelope. See how it's a double membrane? See these little openings in the nuclear envelope? What would those be? Those would be the nuclear pores. So we already have quite a bit that we've talked about shown. Now this blue is the cytoplasm. So this is the liquid environment of the cell. Here is the Golgi Apparatus. See how it's like a bunch of stacks of these membranes? And you can see how they did the membranes here. They filled in the yellow, and they have the red line would be kind of like where the membrane would actually be. And then here's the beautiful part. These are the vesicles. Some of these vesicles are leaving the Golgi. Some of these vesicles are arriving to the Golgi. See, that one's actually fusing with the Golgi right now. Some of them are splitting off of the Golgi right now. And here's the part I want you to really kind of imagine. How are these vesicles moving through the cytoplasm? Remember, they're moving via motor proteins along microtubules. So I can't see the microtubule, but I know it's there. And I know that vesicle is being carried along, just like in that animation, right, with the little guy walking along. It's being carried that way. Now here's the part that's really going to blow your mind if you think about it. This is a two-dimensional picture of a three-dimensional world. So right now what's actually happening at this Golgi Apparatus is from all different directions, vesicles are arriving, and from all different directions the vesicles are leaving. Some are going this way, some are going that way, some are going up towards us, some are going down into the picture, and so forth. And so all around the cell, these vesicles are being carried to deliver their specific packages. So the vesicles are the packages. The Golgi Apparatus is like the post office that receives those packages, modifies the contents of the packages, and then ships them on their way. Next organelle is a lysosome. Now lysosomes are vesicles that contain digestive enzymes that can break down worn out organelles or anything else that's coming in that's maybe not belonging in the cell. Now I like this picture a lot, that's why I gave it to you on your study guide. Number one, it shows us what vesicles look like. Remember, vesicles are these three-dimensional spheres made of lipids, right, membranes. Anytime, by the way, that I've said a membrane like a membrane-bound organelle, realize it's the same kind of membrane that our plasma membrane is made of. It's a phospholipid bilayer. So it's a sphere of phospholipid bilayer with an opening on the inside that you can put stuff. That's what a vesicle is in general. Now, lysosomes are special vesicles. What they have on the inside is these nasty digestive enzymes and acid. Make a note that there's acid as well, right? So there's also acid, enzymes, and acid. And it can break down worn out organelles as well as food particles, and also, make a note of this, also invaders. So maybe it's something that's invaded the cell, like a virus, or maybe it's a cell like a white blood cell that's job is actually to go around and destroy invaders of the body. They have lots of vesicles because they're going to be destroying things. Now, they're found only in animal cells. So if you're doing an animal cell, I better see lysosomes, and if you're doing a plant cell with your project, I shouldn't see them. They should be a little bit larger than the other vesicles, maybe a different color so they're easier to spot, but you should have lots of vesicles, lots of lysosomes in your project. And remember, the vesicles, don't forget, little side note, vesicles should be attached to microtubules as well. Here's a picture showing how this works. You've got a worn out organelle, so some old mitochondria that isn't working so well. And so what happens is it fuses with a lysosome. Now, how is it able to fuse? Well, think about it. Like that last picture showed, the outer part here is just phospholipid bilayer. Well, so is this. So when they bump into each other, you can fuse them together. Now those digestive enzymes and the acids will completely digest this mitochondria. Anything that's reusable, like maybe the amino acids or building the proteins, maybe the fatty acids or building lipids, those can all be released and reused to build a new organelle or something else the cell might want to build, and all the bad stuff gets released. Now, a little side note here. If this is a unicellular organism, then fine, we're done. We've released the waste outside of the unicellular organism. But what about something like me? Well, maybe this is a cell in my pancreas, let's say, and now this waste has been released into my bloodstream. Well, now that waste is going to be filtered out by my liver and my kidneys and gotten rid of from my body. Right? So it's kind of another step there, kind of interesting. Vacuoles. Now here's another one where this is technically not 100% correct. There actually are lots of vacuoles in animal cells, and they do lots of really cool things. Like there's some that actually have iron in them, and they align with the magnetic poles, the magnetic field of the Earth, and we believe that's how a lot of these animals are able to navigate, like birds and butterflies and things like that, using the magnetic field. But what I'm really talking about here is what we call the central vacuole, and it's only found in plant cells. So yes, this is technically not true, but for this level of biology, it's fine to not worry about the small percentage here. And I want us to talk about the fact that plant cells are the only cells that have vacuoles. Now, what's a vacuole? It's a membrane-bound storage compartment. It's essentially a huge container that we can store water, salts, sugar, enzymes, waste, anything that we want to hold on to, either because it's something good or something that we want to kind of store away because maybe it's not the best thing for us. Found only in plant cells. At least the central vacuole definitely is, right? So make sure you know that on the test or whatever. I'm going to say that vacuoles are found only in plant cells. Now notice that the central vacuole fills the cell quite nicely, and as a result, as it's full of water, it's kind of pushing against that cell wall. So it's going to be really important with giving it structure. One thing that's important about this central vacuole in plants that you need to know is something called turgor pressure. I'm going to explain what turgor pressure is. You need to know this for the exam for sure. So this central vacuole is so large that as it's filling with water, it essentially exerts an outward pressure on the cell wall, and that helps to keep my cell nice and rigid and strong, and it keeps the plant nice and vertical. However, imagine you haven't watered your plants enough recently, and so the water starts to leave that vacuole and the vacuole starts to shrink. What happens to the pressure it's exerting? Well, the pressure drops. The cell becomes more flaccid, not nearly as strong, and so not as rigid, and so it sort of gets weaker. And what happens to my plant? Well, my plant shows me that it needs more water. It starts to wilt. It starts to droop. And of course, if you get it early enough and I pour water, what happens? Well, the water is absorbed by the roots, goes into the cells, refills my vacuole, and the pressure is restored, and my plant goes back to looking like this, and hopefully I've caught it in time and my plant hasn't died. That's why plants droop when you don't water them. It's because of this turgor pressure that's created by this central vacuole of each cell. The last thing in this video that we're going to talk about is something called protein secretion. To secrete something, think of like sweat. Sweat is a secretion of your skin. So what does that mean a secretion is? It's something that's released. So protein secretion is how proteins are released from the cell. It starts off with the ribosomes, because we know that ribosomes are what are building proteins. So it's the ribosomes on the rough ER. Those ribosomes produce the proteins. On the rough ER, they're packaged into vesicles, which go to the Golgi apparatus, and where the vesicles are our little containers, our little packages that go to the mail center, the Golgi apparatus. Then these proteins are repackaged into more vesicles that go to the plasma membrane, and as the vesicles fuse with the plasma membrane, the proteins are released. If you can illustrate this on your cell model, you'll get an extra bonus point or two. If you want to kind of remember what this looks like, if you go back to the first video and look at the animation at the beginning, they show protein secretion towards the end of the video. Here's a good picture of what it looks like. We've got the ribosomes on the surface of the rough ER. They're manufacturing the proteins. The proteins are now packaged into vesicles, which go to the Golgi. They then are modified in the Golgi, packaged into new vesicles, and they go to the plasma membrane where they're released. You have this picture in your study guide. Good picture for you to have kind of as a reference. It talks about what's happening, shows you the pictures again. You have the ribosomes manufacturing the proteins, right? The proteins are then packaged into vesicles and go to the Golgi, vesicles to the plasma membrane. That last step, right, the vesicle fusing and releasing it, that last step is known as exocytosis. Think of exit. The word exit means to leave. The vesicle with the materials inside of it. Remember these vesicles, all these membrane-bound organelles, the membrane is the same as the plasma membrane. It's a phospholipid bilayer. So it's able to fuse and become part of the plasma membrane, and what was inside that vesicle is now released outside. You can also bring stuff into the cell using a similar but different process called endocytosis. Think of enter. This is where something that's outside the cell is pinched off into a vesicle, and that's called endocytosis. So I hope this video was helpful with all the different organelles that you'll need to know, and I hope it was helpful as you build your project. I'll see you next time.